Alright, so picking back up where we left off. Alright, so we're talking about antimicrobial resistance to drugs. Alright, so specifically bacteria. Alright, so let's look at the effects of stopping the use of your antibiotics before you run out. Alright, so day 0, alright, you're diagnosed and you're prescribed antibiotics. Alright, so within your body, you have a population of bacterial cells. You have one organism, one specific organism, in this case, causing your symptoms, hopefully, just one. Alright, within that population, you have organisms that are highly sensitive, you have cells that are highly sensitive, you have individuals that are intermediate to the drug in question, and those that are highly resistant naturally. Alright, so this is natural and or acquired resistance. Alright, so we're talking about vertical and horizontal gene transmission. Alright, so after 3 days of taking your antibiotics, notice that your highly sensitive organisms are reduced in population numbers. Alright, your intermediate resistance has lowered, and some of your highly resistant is also decreased. After 6 days, alright, your highly sensitive and intermediates are reduced, and you're left with mainly a population of resistant organisms. Now, if you stopped your antibiotics prematurely, alright, your highly resistant population of cells will begin replicating. And so you'll end up with the overabundance of resistant cells with a particular strain built up that can spread to other people, the environment, and this resistance mechanism can be spread to other bacterial strains as well through vertical and gene transmission processes. Now, if you take your full course of antibiotics, the end result is that you've reduced the population numbers to basically 0, and your immune system will take care of the rest. Alright, so what's going on here? Alright, so you have a colony of bacteria, and one of these strains, one of these cells, has a mutation. Alright, that allows it to be resistant or be more resistant to this antibiotic in question. So if you stop taking your antibiotic, those cells begin replicating and get a resistant colony. Alright, so here looking at process of conjugation, so horizontal gene transfer. Your resistant strain transmitting, for instance, let's say it's a R plasmid to another microbial cell through conjugation. You have transmission of antibiotic resistance to another bacterial cell of the same species, or it can be even closely related, belong to the same genus or even family of bacteria. Alright, so looking at drug resistance patterns. Alright, so looking at the prevalence of penicillin, tetracycline, fluoroquinolone resistance, and gonorrhea from 87 to 2011. Alright, so for some of these, it's been pretty static across the board. So tetracycline resistance has been pretty steady. Penicillin resistance went up, flatlined, and then kind of dropped back down, as back on the rise. And then fluoroquinolone resistance has picked up since the early 2000s and has kept going up. Alright, so we're seeing an increase in cephalosporin resistance as well. Alright, so from the mid-2000s, 2009, 10, 11, you're seeing a spike or increase in cefixime, cephalosporin, and ceftriaxone susceptibility. Alright, so where exactly is this antimicrobial resistance taking place? Alright, well, it could take place out in nature. Alright, so Think about it. Where do organisms come into contact? Where do bacteria come into contact with these antimicrobial agents? Well, think about it. All right, these are agents that are produced by other organisms to fend off competition for resources. All right, so they're already there in the environment. All right, you have in the soil. All right, you have basically viruses that could contain resistance genes or resistance gene fragments for antibiotics or heavy metals or other virulence factors. All right, so looking at the medical side of it, you have the pharmaceutical production of antimicrobial agents, the rampant prescribing of antibiotics for any cough, sniffle, or sneeze, and just our own use of antibiotics. We don't properly use them. We don't complete the full course of treatment. We stop when we start feeling better. And then in agriculture, all right, putting antibiotics into the feeds of organisms, or spraying antibiotics. All right, so you could have from fields, you could have runoff into ponds or lakes or streams. You could have wind spreading these agents around. You can have, again, transmission from other humans, from one human to another, from human to animals, animals to humans. Alright, so here, looking at how this process of antibiotic resistance spreads. So you have animals get antibiotics and develop resistant bacteria in their guts through antimicrobial feeds. Alright, drug-resistant bacteria can remain on the meat from animals. Alright, when it's not properly handled or cooked properly, you can spread transmission to humans. You can have fertilizer or water contaminating animal feces. Drug resistance is used in crops, spread to crops on the farm. It can remain on the crops. We eat it. These bacteria can basically colonize our gut. All right, here you have little George gets antibiotics, develops resistant bacteria in his gut. George stays home and general community spreads the resistant bacteria to other individuals. He doesn't properly wash his hands after going to the restroom, so different types of either indirect or direct contact, he spreads his germs. Let's say that he acquired this at the healthcare facility, so this was a hospital-acquired infection. You could have resistant bacteria spread from patient to patient through improper aseptic technique from nurses and physicians in the hospital. Alright, so individuals going from room to room aren't washing their hands between rooms, touching patients, not changing gloves, things of that nature. Alright, so transitioning to our eukaryotic microorganisms. Alright, so a fungal cell wall. Alright, so fungi. Remember, fungi have chitin in their cell wall. Alright, and then they also have these sugars called beta-glucans, and then they have proteins, sugar proteins, manoproteins. Mannose is a sugar protein. Alright, and they have a lipid bilayer, and this lipid bilayer, they have a gosterol. Gosterol is a steroid, which is a type of lipid. In animal cells, we have cholesterol. Fungi have ergosterol. And basically, they take the building block, squalene, and use that to synthesize ergosterol, which gets inserted into the membrane of the fungal cell. Alright, so looking at antifungal targets. Alright, what can we target if we're dealing with a fungus cell or a fungus? Well, we could specifically target the beta-glucans. Alright, we could target the cell wall of the fungi. Alright, we could basically disrupt beta-glucan synthesis. We can use some type of reagent that intercalates into the membrane. Alright, it disrupts the selective permeability of the fungal membrane itself. So it targets the ergosterol and forms this porous cylinder so that the cell membrane can no longer regulate its internal contents with the external environment. Alright, you can disrupt the synthesis of ergosterol. Alright, so you can prevent squalene from being converted into ergosterol. And then you can also disrupt basically DNA replication. And you can disrupt transcription. Alright, so you can target fungal cell wall synthesis. Alright, so you have polyoxins that disrupt chitin synthesis. Alright, so remember this is one of the many polysaccharides that are found in the fungal cell wall. Alright, you can disrupt glucan synthesis. You can use polyenes that bind to ergosterol and disrupt the entirety of the membrane. You can disrupt ergosterol synthesis. You can disrupt microtubule formation. Alright, so prevent the fungal cells from undergoing a process of mitosis. And then you can inhibit DNA synthesis by using a structural analog of cytosine. Alright, in this case 5-fluorocytosine. It looks like cytosine, but it's not cytosine. Alright, so there's not going to be any complementary base pairing. Alright, so looking at our polyenes. So again, I want you guys to be familiar with the class of drugs. Alright, if you can recall an example of a type of drug that belongs to that class, that'll be good. Alright, so polyenes, you have amphotericin B. So ampho B binds to ergosterol in the membrane. It forms a pore in the membrane by intercalating into the membrane itself, so that now the membrane becomes leaky. Alright, the cell can't regulate its internal environment with the external environment. Stuff just leaks out.
The role of amphotericin. Fungi are eukaryotic organisms which possess a unique cell wall and cell membrane that can serve as targets for antifungal agents. Polyene antifungal agents such as amphotericin B target the fungal cell membrane. The cell membrane of fungi is similar to that of other eukaryotic cells and is composed of a lipid bilayer with proteins embedded within it. A major component of the fungal cell membrane is the presence of sterols, which are virtually absent from all prokaryotes, including bacteria. One of the essential sterols in the fungal cell membrane is ergosterol. In most fungi, it replaces the cholesterol component found in higher eukaryotic cell membranes. Ergosterol provides stability and flexibility to the fungal cell membrane. Amphotericin B is a lipophilic molecule that penetrates the fungal cell wall and inserts itself into the cell membrane. In the cell membrane, amphotericin B exists as aggregates and binds closely to sterols such as ergosterol. This leads to the formation of pores in the membrane, resulting in loss of intracellular potassium and other molecules. Ultimately, fungal viability is impaired and the organism dies. Of note, amphotericin B also has effects on oxidation that may enhance its antifungal activity.
Alright, so your azoles. Your azoles target ergosterol synthesis. Alright, so in this case, you target the pathway that's involved in making ergosterol. Alright, so again, without the ergosterol, or if it's toxic, then, alright, it's lethal to the cell.
The role of azoles. Fungi are eukaryotic organisms which possess a unique cell wall and cell membrane that can serve as targets for antifungal agents. The azole family of drugs specifically targets the fungal cell membrane. The cell membrane of fungi is similar to that of other eukaryotic cells and is composed of a lipid bilayer with proteins embedded within it. A major component of the fungal cell membrane is the presence of sterols, which are virtually absent from all prokaryotes, including bacteria. One of the essential sterols in the fungal cell membrane is ergosterol. In most fungi, it replaces the cholesterol component found in higher eukaryotic cell membranes. Ergosterol provides stability and flexibility to the fungal cell membrane. Lanosterol serves as the precursor for the formation of ergosterol. Although many enzymes are involved in the formation of ergosterol, the 14-alpha demethylase enzyme is responsible for the C14-alpha demethylation of lanosterol. It is an essential enzyme that serves as a target for azole antifungal agents. Although some azoles have an immediate effect of damaging the fungal cell membrane directly, their major mechanism of action is by binding to the cytochrome P450-mediated 14-alpha demethylase enzyme, thus inhibiting the demethylation of lanosterol and blocking the synthesis of ergosterol. In turn, this results in an accumulation of 14-alpha methylsterols and a depletion of ergosterol in the cell membrane. The lack of ergosterol in the fungal cell membrane makes it very unstable. Eventually, the cell membrane begins to break down, and the fungal organism dies.
Alright, so that was a bit of a mouthful. Alright, so just remember that azoles disrupt ergosterol synthesis, and that by disrupting ergosterol synthesis, you don't make any ergosterol. No ergosterol, it goes in the membrane becomes more porous. It's more porous, it's unstable, it's more susceptible to lysis. Alright, your echinocandins. Your echinocandins disrupt beta-glucan synthesis. Alright, so you're disrupting the synthesis of the fungal cell wall.
The role of echinocandins. Fungi are eukaryotic organisms which possess a unique cell wall and cell membrane that can serve as targets for antifungal agents. The echinocandin class of antifungal agents target the cell wall of fungi. In general, 80 to 90% of a fungal cell wall consists of polysaccharides. Glucans, which are polymers of glucose, are the major components of the fungal cell wall, accounting for 30 to 60% of the cell wall of yeasts, such as Candida. The cell wall of filamentous fungi differs somewhat from yeast and include a higher concentration of chitin, as well as in some, a novel linear beta-1314 glucan. A glucan synthase complex located in the fungal cell membrane is responsible for beta-1,3 glucan synthesis. This occurs on the cytoplasmic side of the fungal membrane. Glucan chains extrude from the glucan synthase complex into the periplasmic space where they are then incorporated into the cell wall. Echinocandins act by inhibiting the synthesis of 1,3-beta-D-glucan. Echinocandins may also block beta-1,6-glucan synthesis. This is achieved by binding to the beta-1,3 synthase complex, an enzyme that is not present in human cells. This eventually leads to reduced fungal cell wall integrity, abnormal cell morphology, and finally, cell rupture and death.
Alright, then you can use some type of nucleotide analog. Alright, so nucleotide cytosine analog, so flucytosine. Alright, so here, your flucytosine is going to inhibit DNA synthesis, as well as inhibit protein synthesis. So substitution for uracil. Alright, so by substituting this flucytosine in for other nucleotides, right, you disrupt DNA replication and then transcription and translation of those genes. Alright, so looking at antiparasitic chemotherapy, so antimalarial drugs. Alright, so here, if you're looking at the life cycle of Plasmodium, alright, so here, let's pull up a little video on the life cycle of Plasmodium. Alright, so this is going to walk you through the life cycle of this particular parasite.
When a female Anopheles mosquito penetrates human skin to obtain a blood meal, it injects saliva mixed with an anticoagulant. If the mosquito is infected with Plasmodium, it will also inject elongated sporozoites, motile spindle-shaped asexual cells, into the bloodstream of its victim. The sporozoites travel to the liver, where they enter liver cells and rapidly divide asexually. This asexual division, which is called schizogony, generates the next life cycle form, called merozoites. The released merozoites invade other liver cells and enter the host's bloodstream, where they invade erythrocytes. Once inside the erythrocyte, the merozoite begins to enlarge as a uninucleate cell termed a ring trophozoite. The trophozoite's nucleus then divides asexually to produce a schizont which contains several nuclei. The schizont then divides and produces mononucleated merozoites. The erythrocyte ruptures and releases toxins throughout the body of the host, bringing about the well-known cycle of fever and chills that is characteristic of malaria. Plasmodium enters a sexual phase when some merozoites in the erythrocytes develop into gametocytes, cells capable of producing both male and female gametes. Erythrocytes containing gametocytes do not rupture. Gametocytes are incapable of producing gametes within their human hosts, and do so only when they are extracted from an infected human host by a mosquito. Within the gut of the mosquito, the gametocytes form male and female gametes. The resultant diploid zygotes develop within the mosquito's intestinal walls and ultimately differentiate into oocysts. Within the oocysts, repeated mitotic divisions take place, producing large numbers of sporozoites. These sporozoites migrate to the salivary glands of the mosquito, and from there are injected by the mosquito into the bloodstream of a human, thus starting the life cycle of the parasite again.
Alright, so here you can use antimalarial drugs, alright, to disrupt the life cycle of this particular parasite. Alright, so this is why it's important to be familiar with the life cycle of particular parasites. Alright, so you know how to disrupt, interrupt different stages of its life cycle. Alright, so here we have quinine, chloroquine tablets. Alright, so quinine water. It's got a little bit of an astringent taste to it. So this is a lot of times, initially when it was given to soldiers, this is why quinine in water, basically soda water, would be mixed with things like gin, for instance, to kind of get away from that bitter taste. Alright, so here, alright, the parasite digests the host cell's hemoglobin to obtain amino acids. Alright, so in this case, as a result, you release a lot of heme, which is toxic to the parasite. So in this case, the parasite will polymerize the heme to a non-toxic form of hemozoin, which is sequestered in the parasite's food vacuole. Chloroquine prevents the polymerization of heme to hemozoin. So this accumulation of heme results in both the lysis of the parasite and the red blood cell. Alright, so this particular antimalarial drug is actually derived from genoa tree bark. Alright, so you have other protozoan parasites. Alright, things like Giardia or Entihema histolytica. You have Trichomonas vaginalis. These other protozoan parasites, you would use something like metronidazole. That basically targets DNA and causes DNA damage. Alright, this is basically through oxidative damage. Now, one of the nasty little byproducts or symptoms from using this particular drug is here. You have the blackening of the tongue. Alright, so looking at antiviral drugs. Alright, so your antiviral drugs can prevent the entry of a virus into a cell. Alright, so prevent the virus from binding to receptor proteins. Or prevent the fusion of the virus cell-viral coat with the membrane of the host cell. You can disrupt nucleic acid synthesis, whether it be RNA or DNA, or retroviral transcription of RNA to DNA. And you can also prevent the viral assembly and release of the virus. Alright, so you have things called protease inhibitors. Some of these drugs are basically structural analogs. Alright, so purine, pyrimidine analogs that disrupt viral DNA and RNA synthesis. Alright, so notice here, A is a purine. Alright, B looks like a purine, but it's only a form of molecular mimicry. Alright, C is a pyrimidine. D is a structural analog of a pyrimidine. Alright, so both these particular drugs would be structural analogs that would replace the specific purine or pyrimidine, and it would halt DNA replication and transcription.
Viruses are difficult targets for selective treatment because they rely to a large extent on the host cell's metabolic activities for their replication. Most antiviral agents inhibit viral uncoating, interfere with viral nucleic acid biosynthesis, or prevent the assembly and release of viral particles. After the virus enters a host cell, the protein coat must be removed from the nucleic acid in order for replication of the nucleic acid to occur. Amantadine blocks penetration and uncoating of the influenza A virus. Phoscarnet is an analog of pyrophosphate. It binds to the active site of DNA polymerase and blocks the cleavage of pyrophosphates from nucleotides. It is used to treat cytomegalovirus and other herpes virus infections. Replication of nucleic acids involves additions of nucleotides to the free 3' end of the growing chain. Nucleoside analogs are incorporated into nucleic acids during synthesis, but then prevent further synthesis. For example, azidothymidine, AZT, an analog of thymidine, becomes phosphorylated in the cell and is then incorporated into nucleic acid by reverse transcriptase. Since the 3' position of the deoxyribose is occupied by nitrogen instead of a 3' OH, no additional nucleotides can be added. AZT has been used in treatment of retroviral infections. The HIV genome is transcribed into messenger RNA and then translated into a large polypeptide. A site-specific protease cleaves the polypeptide into functional proteins. Protease inhibitors interfere with this protease and prevent production of functional proteins. Protease inhibitors have been used to treat AIDS patients. Some interferons are antiviral. These proteins are produced by an infected host cell. The secreted interferon binds to adjacent cells of the same kind and stimulates production of enzymes that degrade messenger RNA and inhibit protein synthesis in these adjacent cells. Interferon has been used to treat Kaposi sarcoma in AIDS patients, chronic hepatitis B, and hepatitis C infections.
There is evidence that the infectious agent associated with certain brain diseases.
Alright, so that kind of gave you an overview of different types of antiviral agents and what their mode of action is. Alright, I want you to be familiar with the general mode of action. How could you disrupt a virus in its life cycle? Alright, so here you're looking at AZT. AZT is a drug that's used to treat HIV. Alright, so in this case, you have the reverse transcriptase enzymes taking the RNA and converting it into DNA. Now, in this case, the AZT functions as a structural analog as the AZT triphosphate. Well, here you have this nitrogen. All right, the nitrogen is not a 3' hydroxyl. All right, so it prevents the polymerization of that growing DNA chain. All right, you also have protease inhibitors.
This is the translation event whereby a host cell ribosome will move along a molecule of messenger RNA, and the two of these in tandem are involved in the production of viral proteins. These viral proteins, in fact, include the viral enzymes that are synthesized in the form of long precursor proteins. Different types of messenger RNA molecules, in fact, exist in regard to the virus, and these are translated into different types of viral proteins or polypeptide chains. This is the protein cleavage event, whereby the protease enzyme of the virus cuts these long, non-functional polypeptide proteins into smaller individual subunits that indeed are fully functional. This is an essential step in virus replication. The protease inhibitor, or PI, will now attempt to block the function of the protease enzyme. This is the binding of the protease inhibitor to the protease enzyme, an event which is essential in blocking the ability of the protease enzyme to function, and this then results in preventing the protease enzyme from being able to cleave the large non-functional polyprotein precursors into their final functional units. Hence, if we succeed in blocking the protease enzyme, we will simultaneously prevent the virus from being able to fully assemble in the context of new, fully infectious virus particles. The blocked protease enzyme is now unable to cut the long polypeptide chains into the smaller individual subunit proteins. Hence, the protease inhibitor, or PI, will block the process of protein cleavage and prevent the formation of new viruses. This then results in the arrest of virus replication and clearly is a step that is essential in preventing the formation of new infectious virus, and obviously is a step that has been successfully targeted through the development of new inhibitors of HIV replication that is essential to the success that we have achieved in the therapy of HIV disease.
All right, so here you have this immature viral particle. You have inhibited protease. Inhibited proteases prevents the release of individual core proteins. Basically a full maturation of that virus. Alright, so here you have HIV. You can prevent its binding to host cells. You could prevent its uncoating. You can prevent its conversion from RNA to DNA. You can use drugs that inhibit transcription. You can use drugs that disrupt protein assembly, so it's a maturation of the virus, so protease inhibitors. So there's all different ways you can halt a virus in its life cycle, either at the beginning or the end, or somewhere in between. Alright, so now we're going to talk about some of the consequences. All right, some of the choices you have to make when thinking about what type of antimicrobial agent you're going to use. All right, so you may have an issue with drug retention problems, where that drug gets eliminated by the body pretty quickly, or it's inactivated by a host, or it may even become potentially toxic to a host through bioactivation. You may have drug delivery problems where it's either destroyed or you have poor uptake in the intestines, into the bloodstream. You may have poor diffusion of some type of topical agent through the skin. You could have the wrong drug. You have the wrong spectrum of drug of activity. You could have very few targets that allows that drug to be different from a host cell. Alright, so one of the problems with treating fungal or protozoan infections is the fact that there's a lot of similarity between fungal protozoan cells and our cells. So the drugs are not going to be as selectively toxic because they could be potentially toxic to us as well. As well as the development of allergic reactions or disruption of our normal microflora, all right, which we'll get to. All right, so which of the following antibiotic modes of action will have the least toxic effect on a human cell? All right, so antibiotic A acts on DNA replication. Antibiotic B targets the cell membrane. Antibiotic C acts on peptidoglycan. Well, both of these are going to be very similar to a human cell. This one you're only going to find in your bacteria. Alright, so this is a whole lot more specific. Alright, so looking at choosing antimicrobial drugs. Alright, so you have an elderly alcoholic patient with pneumonia caused by Klebsiella, and it's complicated by diminished liver kidney function, requiring parenteral drug administration. And this patient also has a history of allergy to penicillins. Alright, you have a cancer patient with severe systemic candida infection. Alright, for the first patient, we would use something like a quinolone. So they're allergic to penicillin, penicillin derivatives, carbapenems. So if your cell wall targeting antibiotics won't work. Alright, so in this case, you use something like quinolone. Your cancer patient with systemic candida infection, you probably want to use something like a fluticetazine, some type of nucleotide structural analog in combination with amphotericin B for systemic infection. Alright, so looking at these particular drugs and telling whether or not it's antiprotozoal, protozoal, antihelminthic, alright, it's antiviral, antibacterial, antifungal. So amphotericin B is antifungal. Alright, and bendazole is going to be antihelminthic. Alright, metronidazole is antiprotozoan. Ribavirin is antiviral. Oxacillin is antibacterial. Alright, so one of the other complications of antibiotics is potential allergy. Alright, so in upcoming chapters, we're going to talk about the immune system and allergic responses. to foreign antigens. This kind of gives you a nice little preview. Alright, so here we have our little B cell, our little lymphocyte, and it comes into contact with some type of allergen. Alright, for instance, ragweed. Alright, and this stimulates the B cell to produce antibodies to that specific antigen. Alright, that particular molecule found in or on the ragweed itself to stimulate the response initially. Alright, so here he starts secreting an antibody, alright, IgE, and these IgE antibodies attach themselves to mast cells. Alright, so mast cells are important in allergic reactions. Alright, so now you have mast cells floating around with antibodies attached to them, specific to the ragweed. Alright, so you come into contact with the ragweed a second time. Alright, so this is a secondary contact to the same antigen. Alright, so now the ragweed binds to the antibodies and surface the mast cell. The mast cell will then degranulate, releases histamine and other mediators of an allergic inflammatory response. And this is when you start wheezing, right? Having difficulty breathing, you sneeze, you have runny nose, watery eyes, you can begin to itch, you may develop a rash on the surface of your skin. This is all due to the various mediators that are released by cells of our body, skin, subcutaneous tissue, and inflammatory cells. In terms of something like penicillin, alright, so your penicillin derivatives, alright, what happens is they become antigens, alright, because a host cell carrier protein will bind to the penicillin, forming penicillyl oil, and this becomes what actually stimulates the allergic response.
Anaphylaxis is a systemic reaction that can occur when a patient is allergic After the first injection, a second dose is indicated. A second dose of epinephrine may be essential to adequately counter the anaphylactic reaction. The understanding of anaphylaxis as a continuum, with the potential for a severe, protracted, or biphasic event developing after the initial phase, underscores the need for 2 doses of epinephrine to be readily available for patients at risk.
Alright, so looking at another consequence of use of antibiotics. Alright, so aside from some type of allergic reaction, alright, we have here something called a superinfection. Alright, so in this case, you have organisms that are living in your gut, alright, that are part of the normal microbiota of your body. And some of these organisms are commensalistic. Alright, they don't really pose you any harm. Other organisms are mutualistic in the sense that they're providing you with some kind of byproduct or nutrient of their own metabolism, and you're providing them with sustenance and food, alright, through your diet. What happens is when you take these antibiotics, you disrupt your normal flora. So in this case, you can eliminate your normal microbiota, and you're left with potential pathogens that reside in your gut that now there's no competitive need for resources. They have free reign. Alright, so because your normal microbiota keeps these pathogens in check. Alright, now they have free reign to populate. And so now you have a development of some kind of superinfection. Alright, so here you have an example of a superinfection that could be due to a yeast, for example. Alright, so something like Candida albicans. Alright, for instance. Alright, so in terms of the urogenital tract, this is a typical common normal flora of the urogenital tract. You take a course of antibiotics. We eliminate your normal flora, and these yeast can overpopulate, and you get a yeast infection. Or you could end up with some kind of cutaneous infection as well, whether it be yeast or be bacterial. Alright. Or here, in terms of your gut, in terms of your large intestine, you could have C. difficile that can overpopulate your large intestine. It begins destroying intestinal cells. These are toxins, and the result being a formation of these pseudomembranes. Right, so you see these little yellow polyps. Right, so these are pseudomembranes that are forming, and causes pseudomembranous ulcerative colitis. So colitis referring to some kind of inflammation of the colon. Ulcerative, you have development of ulcers and development of these pseudomembranous structures. Alright, so here you ingest vegetative cells of Clostridium difficile in your diet, or spores. Alright, they make their way through the stomach. Most vegetative cells are killed off. Some may survive. Your endospores will then germinate and make their way to your colon. Well, where they will begin to secrete toxins that will destroy colonic epithelial cells. And as a result, you have the migration of various immune cells to that site, the secretion of immunological mediators that are trying to kill this invader and neutralize the toxins and clear up these damaged epithelial cells. And what happens is you wind up actually losing these epithelial cells through a process of apoptosis. You have the development of this pseudomembrane structure of fibrin coating this area, which is trying to essentially wall off or seal off this infection. And as a result, it leads to inflammation, the pseudomembrane formation, and diarrhea. So here you can see the formation of these little polyps on the surface of the colon.